Go out tonight. We're here today to talk about Venus. If you go out in the night sky tonight or the next clear night, you'll be able to see Venus shining very, very brightly. Venus is an incredible planet. It has many, many, many mysteries. A lot of them are left over from decades ago when the former Soviet Union sent over a dozen probes into the atmosphere of Venus, and the USA's NASA Pioneer Venus also was a multi-probe mission to Venus. So I'm going to tell you some old things about Venus and some very, very, very new things about Venus. I hope I can change your mind about Venus as a potentially habitable world. I'm starting out with a list of what we call atmosphere anomalies, unusual puzzles in the Venus atmosphere that are unresolved, some for decades. This picture is showing you a cartoon of Venus surface, or Venus rather. On the left, we're seeing altitude above the surface. So it's just starting out here at 31 kilometers, going up to 70 kilometers. Now, at the top of Venus, some of you may have heard that surprisingly, half of all incident sunlight on Venus is absorbed in the cloud tops. And it's dubbed the UV absorber, but people don't know what it is. There are many ideas and contenders, but the exact nature of this particle is not yet known. I actually listed these anomalies. It's my personal opinion of the community's opinion on the order of robustness of these ideas. So this UV absorber, definitely there, definitely doing a lot of absorbing. Going down into the atmosphere, also agreed upon, is that if you just use like a basic chemistry, photochemistry model of the Venus atmosphere, you'll find that in reality, there's a depletion of sulfur dioxide and water vapor in the cloud layer. And this is a substantial issue. Continuing to go down, there are some of the cloud particles on Venus in the lower layers. They're quite large, around 8 microns instead of 2 microns of the rest of them. And they appear to be aspherical. We really are curious about these particles and what might drive them to be so different from pure sulfuric acid, which is actually a liquid and spherical in the Venus atmosphere conditions. There are traces of oxygen, which also is unexplained, at tens of parts per million. And there's also indications of ammonia, and many of you, I'm sure, have heard of the reports of phosphine on Venus. So all in all, the atmosphere is mysterious. It holds many unusual things going on, most of them from past in situ missions to the atmosphere of Venus. And so, it definitely lends the question about what is out there. Now, most of you also know that the surface of Venus is too hot for any life as we could imagine. Due to a massive carbon dioxide greenhouse atmosphere, Venus is just incredibly hot, 700 Kelvin at the surface. But just like on Earth, if you hike up a mountain or go in an airplane, it gets colder and colder above the surface. And that's also true on Venus, so that up in the clouds, 50 kilometers above the surface, it is the right temperature for life. Not as important, it just happens to be the same pressure as here on Earth. So you could be in the Venus clouds and have the same temperature and pressure we have here. On our planet Earth, we have life in our clouds. Bacteria get swept up from the surface and goes into the clouds and stays there for about a week. Some of it, much of it goes into the cloud droplets. Eventually it rains back out. So I'm kind of selling here, right, that perhaps Venus has an aerial biosphere. Now, unlike on Earth, where our cloud, I don't know what it's like where each of you are. Here in Boston, it's quite cloudy today, actually. But some days are perfectly clear for days on end. Unlike on Earth, where our clouds are fragmented, Venus's clouds are permanent. They cover 100% of the planet. They're vertically extensive, 20 kilometers thick, and they're always, always there. So, wow, I mean, this idea of life in the clouds seems kind of like it might be promising. And perhaps that's why that over half a century ago, Carl Sagan proposed the idea that there might be life in the clouds of Venus. The one issue that we think he didn't know is that the clouds are not made of friendly water, but they're made of acid, sulfuric acid. In fact, highly concentrated sulfuric acid. And this picture is like a cartoon showing you altitude in kilometers above the surface here on the left. So we're going from 0 to 100. It's sketching out this temperate zone and also showing you where the Venus cloud layers are. There actually is also this lower haze level of unknown material that we could come back to later if you have any questions about it. So concentrated sulfuric acid, I'm not sure if any of you have ever used concentrated sulfuric acid, but it is so dangerous and toxic, it literally destroys all of our life. So I've put things to you. One is temperature and pressure are good for life. There's liquid in the clouds, liquid sulfuric acid. There's an energy source, the sun. So the most bare bones of life requirements are definitely in the Venus clouds. So for some of us who, just as a reminder about Venus, it's about the same mass and size of Earth and same surface gravity. It has a really unusual property that is not related to life in any way, but its day is longer than 1 year. It takes a long time to rotate. And it actually ends up having these incredible winds, and the whole atmosphere super rotates in 4 days. None of this is really relevant to what I'm talking about next. I just wanted you to kind of have some numbers that you could stare at. It's mostly carbon dioxide with a tiny bit of nitrogen. Cloud decks are around 50 kilometers above the surface. Similar temperature and pressure to Earth. Well, that's my introduction for Venus. And now I'm going to move on to the sulfuric acid clouds, because what I want to focus on here is the idea that there could be life in the clouds of Venus. And I'm not asking for your judgment call right now or even later. I'm just going to present to you some amazing and astonishing features of sulfuric acid. And we're going to start with this concept of why sulfuric acid is fatal to our life. And that is because our DNA, shown in this little cartoon here, I like to think of our DNA in 3 parts. The ladder, ACGT, like adenine, thymine, guanine, cytosine. The backbone, and the linker. And the linker is a sugar molecule. So this figure helps illustrate why our DNA is unstable, because sugar is just completely destroyed in sulfuric acid. I'm going to show you a video now.
This is just a quick demonstration to show the dehydrating power of concentrated sulfuric acid. I have warm concentrated sulfuric acid.
Do not use gloves. If anyone works in the laboratory with sulfuric acid, I don't know how this person is not wearing gloves.
Small beaker.
And not regular gloves, but like heavy duty acid resistant gloves. So he's pouring sulfuric acid into sugar. Now watch. Wow. And you don't need to heat it either to do this. You don't need to heat it to see this effect. And it's just absolutely crazy what happens to sugar and sulfuric acid. And this isn't enough to convince us that life can't survive in sulfuric acid. I don't know what is. We think this is why planetary scientists have just assumed for the last decades that sulfuric acid is not only bad for life, but I get this impression that people have been thinking sulfuric acid is sterile to any interesting chemistry whatsoever. So I'm now going to move on to a story of how I got involved in working on sulfuric acid. It started with phosphine on Venus, which I'm happy to answer questions about that later, but because my talk is not about phosphine on Venus, I'm not talking about it now. But the discovery of phosphine on Venus led me to work on Venus, because phosphine is a gas on Earth that is only associated with life. On our planet Earth, there is very little phosphine. It's only made by us humans as insecticides or associated with life in extreme environments. A tiny, tiny, tiny amount could possibly be made in other ways, but it just not in any significant quantities to speak of. We showed on Venus that volcanoes, lightning, meteoritic delivery, anything you could think of just doesn't generate significant amounts of phosphine, even in tiny levels. So I decided to work on the idea of sending missions to Venus that could search for signs of life or life itself. And this started during the pandemic summer. Some of you will remember what a rough summer it was. I had 2 kids at home, including a 17-year-old. There was nothing to do, no jobs for kids, no The sports fields were closed, like they literally wouldn't let you on the soccer field, the basketball nets were gone. So I put my son to work on the Venus project. He wanted something to do, and it was just kind of an online thing, researching engineering materials resistant to sulfuric acid. Well, he finished that so quickly, he pushed to do real experiments. Well, it turns out you cannot order a lot of you can't order a lot of sulfuric acid to your house, but you can order a lot of little bottles. So we ordered a lot of little bottles, and on the left there, this is 2 years later when it's more sophisticated, but look at, this is my summer porch, summer day outside, and he pushed to do real experiments. So I want you to imagine you're a 17-year-old, it's the hot pandemic summer, not a whole lot to do. You're outside on your porch with this laboratory you and your mom have created. You want to put something in sulfuric acid to get started. What are you going to put in sulfuric acid? Something around you, you're just looking around, you're 17-year-old, super bored. You can put it in the chat. I'm just looking for an answer. What are you going to put in sulfuric acid here? It's like, well, okay, well, Mentos water. It turns out water is really bad to put in sulfuric acid because it heats up and goes bad. You are a really friendly audience. Meat, okay, you could try meat. Well, he put an ant in sulfuric acid. And before I could say, please don't do that, this ant literally had a seizure and turned to goo instantly. And honestly, I'm the type of person, I rescue spiders from my house, so I'm not cool with destroying life, but I just want you to know that life seriously is destroyed instantly. But yeah, he did try some other things. He put a rhododendron leaf. It's a very waxy leaf. That actually did okay for a while. Wax from his braces, graphite from a pencil. It turns out there's quite a lot of things that are not destroyed in sulfuric acid. So fast forward a couple of years, we actually started a legit project in our home chemistry lab. And a colleague of mine from Harvard University was retiring, what you see in the middle here, a UV vis spectrometer. That's for my astronomy friends on the call. You put the sulfuric acid in this little cuvette, a little like rectangular tube, and you put your compound into the sulfuric acid. You put it into the machine and it sends UV vis light through. And we actually started to measure a bunch of compounds. I'm going to get into them in a second. And we started to find that some of our life's biomolecules are stable in sulfuric acid. Now, this UV vis machine isn't enough. It was enough for us to sweep, you could imagine doing something cheap and easy in your home lab. But like many projects, you find an answer right away, but the initial work takes many more months to confirm. So eventually, through our friends at Harvard and then on my own, we learned how to do NMR, nuclear magnetic resonance. All our core team became experts on it, actually. And I'm just showing you a picture of this shared facility at MIT. It's in a sub basement, so not the basement, but a lower level. These are all NMR machines. You put your samples in the top here and learned how to do a lot of NMR. So I just wanted to walk you through this for a moment, that even though our DNA is unstable, the ant died instantly, the sugar in the DNA molecule, but our first experiment was on our nucleic acid bases. We just literally ordered the compounds of pure adenine, thymine, guanine, cytosine, and a few others. We put them in sulfuric acid and we just measured their absorption curve. So this is showing you absorbance from 0 to around 1.5. And the bottom is wavelength in nanometers. And you can see that after just as long as it took us to get it ready, which was about half an hour, that's the blue curve. And then after a day, we remeasured it. That's the dash curve. Your job isn't to understand all the details here, but ask yourselves, are the two curves the same in each picture? Dashed versus solid. The answer is yes, they are the same. Over a day, a little more compound dissolves, so we get a bit of a stronger signal after 24 hours. But essentially what we saw was that the compound didn't change in a day. And that's very significant because it indicated to us that the molecules, our DNA nucleic acid bases are stable in sulfuric acid. But this is not enough to elucidate the structure. So eventually we had to go and do the NMR. And this I'm going to leave you to read in the paper because I want to move on to another topic, but I'm just showing you the picture on the right. Now these nucleic acid bases, they're getting protonated, but they actually, because an acid wants to donate a proton, but they've been stable, almost too stable, because after a year or 18 months, I keep the tubes here, the NMR tubes, and we just keep trying it every once in a while, and they're just actually stable pretty much indefinitely. Well, I just want to tell you more about the amino acids. We took our 20 biogenic amino acids, and I just have a picture here for those of you who know something about organic chemistry. For the rest of you, just think of it like a pretty picture. Again, we just ordered them like a set of our 20 biogenic amino acids, and we put them in sulfuric acid to see what would happen. And I just have like one sample slide on NMR, and this is alanine. And what you're seeing here is intensity. It's dimensionless. It doesn't have like a unit. And here you're seeing this part per million shift. But what's happening here is it's in a tube of compound dissolved in sulfuric acid in the NMR machine. The NMR machine's a giant magnet, so it's making a magnetic field, and it's sending radio frequency pulses to excite the nuclei, and then they relax back to their normal position and give off a signal. Each atom, and this is carbon NMR, 13C carbon NMR, so each carbon atom has to have its own peak because how much it gets affected depends on the environment of all the electrons surrounding it. For your purposes, you just have to see there's 3 carbon atoms indicated by the 1, 2, 3. And for my astronomer friends here, honestly, it's insane, but in organic chemistry, they don't even draw the carbon. You know, like it's the first thing you have to know if you're going to go to astrobiology talks. It's just, okay, I'd rather them draw the carbons in, but there's a carbon at each of these positions, and that's 1, 2, 3. And it takes some time to understand why they're shifted. We're not going to go into that here. What your job to look at is the different colors. Black, that's 98% concentrated sulfuric acid. Yellow is 81% concentrated sulfuric acid, the rest being water. And the red is water. And so look, they're all overlapping. If the peaks are all overlapping, it means there's no change. And this is how we're proving that this amino acid is stable in concentrated sulfuric acid, because 1 week, 1 month, we measure it, and these peaks do not change. That's how we elucidate the structure of this molecule. We're comparing it essentially to how it behaves in water. If you wanted to know why we're using D and not H, it's just because we also run HNMR, and we don't want the H2SO4, the H from H2SO4, to overwhelm our sample. And so we have our background solvent being the deuterated version. So we put all of our amino acids in concentrated sulfuric acid. For all the black molecules here, they're essentially unmodified and stable. The ones with blue or red are chemically modified. So we find some molecules are sulfonated or sulfated. And then there's one molecule, tryptophan, which is kind of fragile to begin with, that is not stable. And again, this plot, everyone will understand it, but we wanted you to at a glance, even if you're not understanding any organic chemistry, just to take a look at all these 20 biogenic amino acids and see that, you know, half of them are modified, but after modification, they're stable in sulfuric acid. Okay, some of you might ask, okay, I'm going to leave the dipeptides for Q&A if you want to ask more about it. But I wanted you to know that my goal that I've imposed upon my team is to generate a DNA-like molecule, an informational polymer. So not our DNA, but we want to show that a kind of DNA can survive in concentrated sulfuric acid by, for example, swapping out parts. We can imagine that if our nucleic acid bases are stable, perhaps we can construct a backbone and a linker molecule that are stable to concentrated sulfuric acid. I'm not trying to create life in the lab here. I'm just trying to convince all of you and potential funders of the missions that there could be life in sulfuric acid. And that means using sulfuric acid as a solvent instead of water. So now everything I've told you doesn't mean there's life on Venus, and I'm not trying to say there is. I'm just trying to say that despite a lot of biomolecules being unstable in sulfuric acid, it appears that a lot are stable. And we want to really try to explore this idea further. Now, if you're all paying, those of you who are following this, we do have a little glitch actually that we're working through. And that is that these nucleic acids, this is going a little bit deeper, so you don't have to understand this to understand the rest of the talk. But I was telling you how some of these these nucleic acid bases, they're getting protonated, a hydrogen is getting added on. And that actually might interfere with this ladder that stays together by hydrogen bonding. If we have extra hydrogens attached to these ladder molecules, they may not be able to hydrogen bond. So part of our exploration may have to include alternate base pairs. And I just wanted you to know that there are alternate base pairs out there that should not be protonated in concentrated sulfuric acid. And these base pairs, they're not joining together by hydrogen bonding, but by other bonds, such as hydrophobic or van der Waals interactions. And, you'll have this talk online, or you can read about it later, but we found this really fascinating thing that there are these unnatural human-made synthetic nucleic acid bases, and some researchers actually put them into a life form. And they put these unnatural bases into the DNA of this life form and instructed it to make a new protein. So I'm just trying to tell you that it's possible there are different types of DNA out there in our universe, on other planets in our galaxy, and we're working towards this DNA-like molecule. So I hope you're still with me, because we are proposing something almost heretical, that there could be life elsewhere, on Venus perhaps on an exoplanet, that is using solvents other than water. And we're specifically talking about sulfuric acid here. So I told you about amino acids, nucleic acid bases towards a DNA-like molecule. But we have extended team members also working on this problem. And some of our team discovered that concentrated sulfuric acid, it's not sterile to interesting organic chemistry, as many of us seem to have just adopted from osmosis from our community. They did experiments, for example, putting formaldehyde, a small organic molecule, or carbon monoxide gas, and this picture is showing you a little tiny piece of a meteorite, into sulfuric acid, and they found that it grows into a rich organic chemistry all the way to ringed molecules. They were really surprised to find this. And when they looked into the literature, they found this as a well-known fact, not in astrobiology or planetary science, but in the oil industry, where they use concentrated sulfuric acid to go from crude oil to more sophisticated organic products. We've most recently put out a paper, it's on AstroPH. We put it on the Astronomy Archive, Planetary Science. And this is that if you take these molecules here, if you take lipids with polar heads and people put these into water, in the idea where people want to show how life originated, how life formed in water. And these lipids, they're amphiphilic, but they put the polar head outward and the water loving part outward, the water not loving part inward. And they form these little tiny spherical vesicles around the same size as cells. Now, no one's calling this a cell membrane, of course, because a cell membrane is a rich tapestry of a lot of different things, but it could be conceived of as the first step. Well, we put these into sulfuric acid. And I sometimes like to ask people, do you think my colleagues, Janusz Petkowski and Daniel Dusevic, do you think they were so brilliant that the first molecules they tried made spherical vesicles in sulfuric acid? Or do you think a lot of compounds could do the job? Well, I hope the answers are both correct, but it turned out we accidentally found in the literature from decades ago, after we had done our own experiments, that there were a couple of groups doing this, not for life on Venus, because back then they didn't know Venus had sulfuric acid clouds, but just for chemistry in the laboratory. So I want you to know that it is my scientific opinion that this is the tip of the iceberg. And there's so much more about sulfuric acid we just don't know, that these just tiny 3 little threads that we hope some of you will take and try to grow it and find out what's going on is really important for us in understanding not just the potential for life in our own solar system, but the possibilities for life elsewhere on exoplanets. Just to close this part of the talk, I'm not saying there's life on Venus. I am saying that it's worth going to Venus to see what's there based on these laboratory experiments. So what are we going to do about this? We're going to go to Venus. And this, we have a consortium that we call the Morning Star Missions to Venus. Right now, Venus is in our evening sky, but it's often in our morning sky, shining brightly. We like to think of this as like a new dawn is rising, not just for opening our minds to think about things way different than here on Earth. But also we want to generate like a new model for space science. It's been nearly 4 decades since any country has sent a probe into the atmosphere of Venus. And so we're trying to change that by working together with new space that's like faster. That's not a CubeSat to Venus, but it's the concept of how more countries, smaller instruments, more off-the-shelf components can be adapted. Just like the CubeSat industry has helped many get into space, we want to build upon like cheaper launches and better way to do space science. So I'm going to briefly talk about that for the last part of my talk. We call ourselves Morningstar Missions to Venus. And our first mission has a more specific name because we've partnered with Rocket Lab, and we call it the Rocket Lab Mission to Venus. I'll give you a brief overview of this mission, targeted for 2025. This mission is like the first of what we plan to be a series. So far, this is the only mission that's funded and being built right now. It's kind of a mission of opportunity. This mission will not find signs of life on Venus. We are aiming to show and confirm that the droplets are not pure sulfuric acid, that their composition or the shape of the mode 3 cloud particles, if it's not a sphere, it's not pure sulfuric acid. This is a very small payload. It's a cheap mission. We have no parachute. We're going to rush down through the cloud layers, only slowing down by our own drag. This is showing you altitude above the surface and time from entry. And just look at this black line here. And we have in the cloud layers of, we just have literally 5 minutes in the clouds. And I wanted to tell you what we're going to be working on. Let me just see what I can say here quickly. Okay, for any engineers here, we have this very small probe. It's like 40 centimeters in diameter. It's probably around 25 kilograms. It is like this kind of half cone, and it has to have a very special heat shield so the whole thing doesn't evaporate. And it has this transparent back layer so we can communicate. What you're seeing here, among many other things, is this instrument. We get our single instrument that's a 1 kilogram instrument, and it is sending an ultraviolet laser through a window. We're trying to send it outside of where the airflow around our probe, because we don't want to accidentally catch particles that were ablated off our heat shield. So we have this 20 centimeter focal length, and we're going to interrogate these particles by sending an ultraviolet laser out and see what comes back. The first thing we want to do is look for fluorescence. And on the left here, I'm just reminding all of you what fluorescence is. It turns out that many organic compounds are known to fluoresce, especially these ringed molecules where the electrons are delocalized. And so we're going to be looking for fluorescence. It won't be 100% sure. It won't tell us what kind of organics are there, but it, again, will tell us that there's something very interesting in the cloud droplets. This right plot is just showing you, we're not going to go over it now, but it was just laboratory experiments where we use different excitation wavelengths, different emission detectors. So we could choose for our small, cheap instrument, we could only pick one laser and one detector. We're also going to measure the backscattered polarized radiation. Just so you know, if you took a light and like shined it at different particles, dust particles, gas molecules, anything, nothing in nature scatters equally in all directions. The way that materials scatter light as a function of angle, and especially also in polarization, has clues as to what the particle is composed of. The former NASA missions did have nephelometers that did this experiment. They didn't have polarization though, and we do. So we're, again, trying to constrain what might be in these cloud particles. That's this first mission. We have our next mission that we are now fundraising for. We call this mission Habitability Probe. We expect it to be a small balloon, bigger than the former Soviet Union's 2 balloons that went to Venus, but nonetheless have a gondola that has one main instrument in the gondola. And this instrument, we intend to be a mass spectrometer. This mass spectrometer is going to be very special and not, fragment all the molecules. It's not going to pyrolyze the molecules like most mass specs that have been to planets before. And we have some partners who are on this call, including from Finland and from Estonia. So great to see you here. I don't have time to go into all the details here, But if all went well and we're confident we're going to get this second mission off the ground, we're aiming for a 2031 timeframe to go to Venus and to investigate these cloud particles and just to find very complex molecules of the kind that only can be associated with life. That's our goal. But further in the future, we want to see our world, our Earth, all the people here, get our act together and send a balloon, a probe to Venus that can bring a sample back. This is kind of way out there, so I'm not going to dwell on it. I just want you to know that it's kind of a natural end goal to want to bring a sample of the Venus clouds back here to Earth and be able to study them in a lot of detail in our laboratory setting. You can read all about this. It's obviously changing with time and material gets outdated rapidly. We used to call ourselves Wiener Life Finder missions. We did receive a lot of pushback from all kinds of people in the community, private, potential private foundations giving us money, to NASA, to a lot of people. So we rebranded Morningstar Missions to Venus. But you can find our 120-page report, and we broke this down into a series of papers in a special issue of MDPI Aerospace 2022, if you want to read more about pretty much most of the things I've talked about. So to summarize the takeaways for today, Venus has many long-standing mysteries. Some are measured directly in the atmosphere by probes from nearly 4 decades ago. These mysteries have to do with the anomalous cloud particles, signs of gases that don't belong, and other aspects of the atmosphere. We are promoting that sulfuric acid, concentrated sulfuric acid particles are not sterile to interesting organic chemistry, based on the laboratory experiments I described to you and touched on our extended team's work. It's a new era for Venus exploration. NASA and ESA are sending missions to Venus, and although those missions will not probe the cloud particles, will not search for signs of life, we're able to instigate and to motivate a series of very focused, specialized, astrobiology-focused, private public funded missions to Venus. Thank you for your attention, and that concludes my talk.